So I uploaded some of the video on the YouTube, and I hope you watched already. And this is the first lab since the beginning of the semester for Tuesday. So we're gonna do 3 jobs today. First thing, we will do a bacteria transfer. Okay, I'm still going to talk a little bit. Bacterial transfer, we mentioned about aseptic technique. So what is aseptic technique? Which means we're going to be transfer an unwanted, let's say bacteria, into a sterile or sterilized medium without carrying unwanted, let's say microorganisms. Well, this is called contaminants. Okay, so that means basically is you loop. You have to be flamed. Make sure it's become red, and then you have to be reflamed between each of the action, each of the transfer. So what we're going to do today, everybody you will see there is a plates in front of you. So there is a plates. There is other plates in front of you. So we're going to do a 3 transfer. Number 1, we will be transfer from the plates to a broth solution. Number 2, we will be transferred from a plates to an agar slant. Now, why we need to do this agar slant? Because we thought it's a short time storage. Okay, it's for the short time storage. This is the one you need to do is we call a zigzag transfer. And this is you just pick something, go to the broth. Okay. Now number 3, what we're going to do, we will do a plates, go to also an agar plate. This will be, we call streak plating. Okay, so how to do streak plating? I did that, but maybe the video you didn't see it clearly. So you pick the colony from here. Let's say you pick E. coli. Use loop. You will do a very intensive streak at the first quadrage. Then you're going to flame the loop. So you have to flame loop. Flaming loop. Then from the first quadrage, do a second streak. That is less intensive. Then you flame loop again. Do a third quadrage, less intensive. Then you flaming loop again from here to do a last one. So that's called the fourth quadrage. Now every time when you do a streak, you have to come in from the before one. Which means you do a second quadrage, you have to come in from the first one. You do the third one, you have to do the coming from the second one. You can't do like this. Okay, lots of you do like this. First quadrage, then now what happened? Every single grow here, they will not have a single colony. Because when we do streak plating, we want to show these single colony after incubation. And this is we call a pure culture. Is that right? That's called a single colony. Okay? You're going to do this work first. This takes you only 5 minutes. Now, the first thing you do, very important, is label. Don't forget the label. If you see the video, I do the label first. If it's a gloss, you're going to be label on the top. If it's a slant, you're going to label on the side. If it's an agar, label bottom. Never ever do is on the lid, because if the lid is gone, then you never know the plates. So what are the information you're going to put it there? Is your name, batch number, of course initial, okay? Batch number, what kind of the bacteria you put it there. If E. coli, you write EC. Bacillus, you write BS. Staphylococcus aureus, you write SA. And then the date. And you can put your batch number there. Okay? Now the bottom line is next Tuesday, you can find your plates. That's very important. Now you're going to give those to TA, and she will do the incubation for all of these. All these plates and the tubes, we will do 35 degrees Celsius, incubation 24 hours. Now, because all the bacteria we're using today, we call it a mesophile. Is that right? That's a good temperature, is about 25 to 35 degrees Celsius. The optimal temperature is 35 degrees Celsius to grow. So this is the transferring bacteria, the first thing we will do. Okay, so the second thing, this is the one supposed to be, we finished for the Thursday lab. We will skip to do the simple stem, because gram stem is cover some steps of the simple stem. So this is the first job. Second job, what you do is gram stem. So you will get slides, which is from here, from the front desk. I'll turn it up, I'll open this one for you, and you will get right here. Everybody get one slice. You have to label. This is the one you're gonna only do E. coli. Okay, we're gonna put E. coli on plates. So we will do E. coli. Here you're gonna draw two circles. EC is E. coli. You do SA, which is Staphylococcus aureus. That's E. coli. And this is Staphylococcus aureus. Then you flip it over. Remember, we have to flip it over, or the dye will damage the lens. Okay, now then what you do, pour one drop of the water, then touch bacteria. Every time when you pick bacteria between E. coli, Staphylococcus aureus, you have to flip it. Okay, then make a smear. Then we do gram stain. What's a major step of the gram stain? Crystal violet. Then gram iodine, followed by decoloration. Use 70, or actually we have a 90% alcohol. And last one, we counter stain, use saffronin. Okay, this is the major steps. Then we dry, and then we do the observation. We do observation is 10x, you find a specimen, 100x. With oil immersion, you will find the three items under the gram stain, which is gram reaction, morphology, and arrangement. These are the three items. Okay, now the third thing, you're going to grab another slide. So we will do endosspore stain. So when you finish gram stain, just left the glass slides there. Then you go ahead to do endosspore. And go back to do the observation. Endosspore, we will use the same smear preparation. We only going to do one. Bacillus subtilis, or Bacillus cereus, or BC. Bacillus cereus. So same thing, you make a smear. Okay, once you make a smear, you need to ask TA, and everybody go back. There is a water bus. Because we have to do is the flooding with malachite green. And then we do a heat steaming. This heat steam here, we're going to use water bus. What I'm doing in the video, I use the Bunsen burner back and forth to do on the surface. Here we'll just use the water bus. I think the water bus is already being heated. Okay? If it's not good, we can go back to use the Bunsen burner to do it. Because it's a very rigid system for the indoor sport. We need to use malachite green heat steam to let the dye go inside. Then we rinse it, we counter stain use saffroning. Okay? So you finish this, of course you use 10x fine specimen, 100x with oil immersion. You're gonna find the indoor spot. Okay, so we finish all this work. Then together with a simple stem, and next Tuesday I'll go over the results. Okay, so that's what we're going to do. Any questions? If no question, go ahead to do. Do the transfer first, then gram stem slides, indoor sport stem slides, then you grab a microscope to do the observation. Okay, that's a procedure. Good, that's all I have.